Everyone. Today we will be talking about Mhc molecules or Major Histocompatibility Complex. What is Major Histocompatibility Complex? Major Histocompatibility Complex is a term which is used in like it is a collection of genes. It's not just one gene. MHC is a collection of genes, and it is arrayed within a long continuous stretch of DNA, and it is present on chromosome number 6 in humans and on chromosome number 17 in mice. So MHC is referred to as HLA complex or human leukocyte antigen complex in humans, and as the H2 complex in mice. So MHC is the region that is actually responsible for determining whether or not any tissue or organ that you are during transplantation from other individual will be accepted or rejected. So we often hear the term rejection of organs, or we often hear the term that your body is rejecting any kind of any blood or any organ, kidney transplantation or liver transplantation. So why is that happening? That is happening because of the MHC complex. So MHC complex, they have other functions, like they are required for antigen recognition by T cell receptors. Now T cell receptor can only recognize antigen which is presented by MHC molecules. So this we have been repeating since first class, that T cell receptors cannot interact or cannot recognize any antigen in its whole form. Antigen has to be processed in order to be processed and then presented on an MHC molecule to be recognized by a T cell receptor. So that there the MHC molecule plays a very important role in immune responses. And MHC molecule is also very important for determining your self and non-self components. So MHC, all your cells, basically all cells of your body will be expressing some types of MHC molecules, which will be different from other person's MHC molecule. So that is why it can determine whether or not a tissue or organ transplanted from another individual will be accepted or rejected. So it can be the difference in MHC molecules can range from 1 to 10%. So if you are getting a quite good match of your kidney or your liver, so then you can go ahead with your transplantation with minimum risk. But most of the cases in most of the individuals will have very different varieties of MHC molecules. So MHC molecules are having two major classes, mainly MHC class 1 and MHC class 2. So we have been talking about MHC class 1 and class 2 in the last class as well, and we had discussed about their different differences in MHC class 1 and class 2. Today we will be studying about its structure and how they are presenting peptides by different exogenous pathway or endogenous pathways. So MHC class 1, as we know, that is expressed on the surface of all nucleated cells. So basically every cell in your body except your RBCs or non-nucleated cells will have MHC class 1. And MHC class 2 is expressed mostly on the surface of antigen presenting cells. So what are antigen presenting cells? Again, just a brush up. These antigen presenting cells include B cells, macrophages, and dendritic cells. So these are the professional APCs. And apart from MHC class 1, class 2, this MHC class 1 and class 2 molecules, these are expressed on the surface of membrane. So they are membrane bound glycoproteins. They are not in the secreted form. They are always embedded on the surface of a membrane. So they will always be embedded on the surface of membrane, and they have similar quaternary structure, but they will differ in the pattern of how they create such shapes. So you will see in the next slide how they are having almost similar kind of quaternary structure. So we know about the primary, secondary, tertiary, and quaternary structure of proteins. So these are found by different protein foldings. Primary is basically the peptide sequence. Secondary is just alpha helix or beta sheets. Then then comes the tertiary structure where there will be some kind of folding. Then comes the quaternary structure where there will be two or three subunits coming together and making a whole protein structure. So class 1 and class 2 molecules also have two subunits to come together and make a structure. So we call it as a quaternary structure, and we'll see how their structure is quite similar yet different. So we have also come across a term that class 3 Mhc molecules. So class 3 Mhc molecules are not as much studied as like class 1 and class 2, and they are unrelated protein molecules, and people have no idea as to what kind of proteins they are recognizing. Mostly they are recognizing lipids, but it is still under research, and also they do not share any kind of structural or functional similarities with MHC1 or MHC2. So all the study that people have done or researchers have done over the years have been concerning just the MHC1 and MHC2, and for studying MHC class 3 molecules, maybe you have to dig deeper into research articles. So the structure of MHC molecule, so as I mentioned, that it's a quaternary structure. So it has it has basically two chains. So it is an alpha chain and a beta chain. So both MHC class 1 and class 2 has an alpha chain and a beta chain. Now the difference in the structure is, in MHC class 1 molecule, you will see there is just one transmembrane domain. But in case of MHC class 2, we have two transmembrane domains. Also, there is so the membrane proximal domains, or the domains which are closer to your membrane. So this is the structure. This is towards the outer side of your cell. This is the cytoplasmic side. So your MHC molecules are expressed. So if this is the cell, MHC molecules are expressed on top of your cells, right? So this is the cytoplasmic part, which is denoted at this end, and this is the outer membrane part. So the membrane proximal region, or the membrane proximal region consists of alpha 3 segment and beta 2 microglobulin in case of class 1 molecule, whereas in case of class 2 molecule we have alpha 2 and beta 2. So alpha chain in case of class 1 molecule has three segments: alpha 1, alpha 2, and alpha 3, whereas in case of class 2 molecule we just have two segments: alpha 1 and alpha 2. Similarly, beta microglobulin has just one segment in case of class 1, and in case of class 2 we have beta 1 and beta 2. So mostly the region which is towards the membrane distal side or towards the peptide binding cleft. So here in case of class 1, this is the peptide binding groove or peptide binding cleft, which is formed by alpha 1 and alpha 2 in case of class 1 molecule, whereas in case of class 2 molecule, the peptide binding cleft is formed by beta 1 and alpha 1. Now this peptide binding cleft, so this region of your MHC molecules needs to be highly variable. So it has to, because it has to recognize a lot of peptides, a lot of antigens. So antigens, not peptides, to be specific. So accordingly, these regions are very variable. So alpha 1, alpha 2 region is quite variable in case of class 1 molecule, whereas alpha 1, beta 1 segment is quite variable in case of class 2 molecule. Whereas the beta microglobulin parts, which are towards the membrane proximal domains and are not interacting directly with the peptide, these are more or less conserved. So this consists of variable regions, this consists of conserved regions in MHC molecule. Right. So accordingly, the structure of MHC molecule, as you see, these are made up of disulfide linkages. These are all the quaternary structures of both the class 1 and class 2 molecule, and you see they have similar kind of bonding patterns in both class 1, class 2 molecules. Just there is some minute differences, like just one transmembrane domain in class 1 and two transmembrane domain in class 2. So apart from that, the structure are quite similar, but also the differences in MHC1 and MHC2 is the peptides that they bind. So class 1 can bind to mostly the endogenous peptides or viral protein, and this class 2 molecules will bind to exogenous peptides or say bacterial infection or any kind of external pathogens. So endogenous peptides includes viruses, exogenous peptides includes bacteria or say fungi, etc. Whatever is coming from outside of your cell. So apart from that, there are certain terms that are associated with MHC molecules, and I found that it could be helpful to explain what does polymorphism or what does polygenic mean. So we in the lecture we have seen that MHC molecules are known as polymorphic. So what do you mean by polymorphism or polymorphic property? So MHC genes are highly polymorphic. That means that they will have many alleles or alternative forms of each gene in a population. What does that mean? Now say in a particular person, we have one gene, gene 1. Now we can have several alternative forms of gene 1. So gene 1 prime, gene 1 double prime. So all these genes, this could be different in within the individual, within the same individual, or could also be different within different individuals. So your same gene, same MHC gene could be hi Khushi. Yeah, I will be discussing practice questions in the last at the end of the class. So polymorphism means that we'll have different alleles or alternative allele forms of the each gene in population. So different different alleles means that different there will be minute differences in particular sequences of this gene. So because of this polymorphism, we can find out that MHC molecules can bind to a variety of peptides, because this antigen binding cleft or the peptide binding cleft of your MHC molecule is highly variable and highly polymorphic, which means that it will have different alleles of the same particular gene. So it will show different binding properties to different kind of antigen. Also, MHC genes will lie very close to each other, so their inheritance is linked. So there are several genes, say MHC gene 1, MHC gene 2, MHC gene 3. So all these genes, they lie very close to each other, so their inheritance will be linked. So all three genes will be inherited at the same time instead of recombination, because most of the times when we are studying any inheritance, what will happen is your genes will get recombined and then you will obtain a set of recombined genes from your parents. But MHC genes, they lie so close to each other that all the three genes will be inherited at the same time in most of the cases. So as it is written, most individuals will inherit all linked alleles encoded by these genes as a set. So all these genes, gene 1, gene 2, gene 3, they are inherited as a set. Now, so this set of linked alleles is known as an haplotype. So we have if a mother has one kind of set of genes, say gene 1, gene 2, gene 3 belongs to your mother, gene 4, gene 5, gene 6 belongs to your father. Similarly, you will have two, so there are two strands basically from your mother's gene and two strands from your father's gene. So only one of your haplotype from your mother and one haplotype from your father will be inherited from your one haplotype from your mother and one haplotype from your father will be inherited during reproduction. So what is the inheritance of MHC haplotypes? It could be explained in terms of an inbred mouse strains. So if you have a mouse with H2B parent, so because MHC is in case of mice, the nomenclature is H2 genes. So here we are using a B type of gene for mouse A, and K type of gene for mouse B. So this is the mother and this is the father, suppose. So if you are mating these two mice, then the progeny that you are getting, F1 progeny, will have or will express both types of MHC molecules. So both B types. So this is one haplotype, B is one haplotype, K is one haplotype. And it will express both B and K in the F1 progeny. So here the nomenclature is H2B/K. And this is another property of MHC molecule, that they are co-dominantly expressed. So when one haplotype from mother and one haplotype from father is coming and recombining in the offspring, so it is not going to be any kind of dominant or recessive trait. Both the haplotypes will be expressed in equal amounts, so which is known as co-dominance, or that is why MHC genes are co-dominantly expressed. Also, this property was again shown in experimentation, where you are using a skin graft donor. So we know that MHC molecules, these are responsible for rejection or exception of your particular transplantation. So what they did is, they are taking this graft from BB parent, orange colored, shown here in orange, and they are taking the skin and grafting it to another parent of the same haplotype BB. Here what happens, the skin gets accepted. Whereas if you are transplanting the skin in the KK type of the parent, it will not be accepted, and it will be rejected. So immune responses will be started against this orange skin part. But if this skin graft from this parent is uh, transplanted to the offspring, it will be accepted in the offspring, because the offspring has the expression of both B and K. Similarly, in the KK parent, if you are taking the skin and transplanting it in the yellow mice, it will be accepted. But if you are transplanting in the orange mice, it will not be expected. It will not be accepted. But if you are taking the skin graft and transplanting it to your progeny, it will be accepted, because it has the expression of both B and K genes. So this is codominantly expressed. Again, here if you're taking the skin graft from the progeny and transplanting in the parents, none of the parents will be able to accept it, because it is again a new type of MHC molecule. So this is again this MHC molecule. So different. So as your breeding goes on and on, your MHC molecule also will become more and more differentiated, and more amount of genes will be mix and match will be taking place, and more newer genes will be synthesized. So another property of MHC molecule is that MHC molecule can bind to a diverse variety of peptides. So it can bind to a lot of peptides. It is not specific to any kind of antigen or any kind of peptide. So this is possible because MHC molecules are evolved in such a way that it can include multiple genetic loci. Means in the MHC gene, you have many genes that are encoding proteins that have same function. Okay, so if there are many genes that are encoding proteins with the same function, then any one of the gene which is going to recognize your peptide, so it will not be a problem in recognizing the peptide, because different genes will still recognize the same peptide. Although there could be some kind of little non-specific pairing, but MHC molecules are not that specific in terms of recognizing any peptides. So here it will allow the binding to more diverse variety of peptides due to this property, and this property is known as polygenic. Polygenic means many genes. So MHC region is therefore said to be polygenic, as it contains many genes which can perform the same functions. So as I was talking about the polymorphism in case of MHC molecules, so polymorphism means the same different kind of amino acid residues which are present in the peptide binding cleft. So on the this figure basically represents all the red colored red colored patches in your alpha helix structure and the beta sheet structure here denotes the amino acid numbers of those amino acids which are polymorphic or can show different, like in different genes it will have different sequences. So this sequences are not conserved, and there will be polymorphism in mostly alpha 1 and alpha 2 domain in human MHC class 1, whereas in alpha 1 and beta 1 domain in case of MHC class 2, because these domains constitute the peptide binding cleft. So also I would discuss in brief about how the stabilization of MHC class 1 molecules is carried out. So basically MHC class 1 molecule, after being synthesized, so it is synthesized in the ribosome and then it will go to the ER membrane. So this is the membrane of your ER, rough endoplasmic reticulum, and here your MHC molecule is expressed, or it is just synthesized, and now it will undergo the assembly between the different chains of MHC. So this is having just in a class 1 MHC molecule, thus the alpha chain is present over here. So the alpha chain is then bound to the calnexin and ERP57. What are these? These are actually chaperone proteins. So what are chaperone proteins? Chaperone proteins are proteins that assist in protein folding, that assist in proper protein folding. So MHC1 class 1 molecule, after being expressed, the alpha chain of MHC1 molecule after being expressed on the surface of the rough endoplasmic reticulum membrane, it will bind to this particular chaperone proteins, and these chaperone proteins will bind to the molecule, and until and unless it will fold properly, until and unless it is stabilized, it will not leave the molecule. So class 1 MHC molecule in its peptide unbound state is highly unstable. So this is peptide unbound state, and it is highly unstable in this state. So in order to stabilize it, what will happen? The chaperone proteins will bind. So once the chaperone proteins bind, there will be another subunit, the beta 2 microglobulin subunit, that will bind to the MHC class 1's alpha chain. So once the beta 2 microglobulin binds to here, so two other chaperone proteins like tapasin and calreticulin, they will again bind to this complex. So the entire complex that will be formed by is ERPP57, calnexin, calreticulin, and tapasin. Binding of tapasin will allow the binding of this MHC class 1 complex with another protein known as TAP. So TAP is a ABC transporter. So now in your cell, in any cell, there will be several proteins or several peptides that are synthesized. Not all the peptides or not all the proteins that are synthesized is useful. Some proteins need to be degraded, some proteins are misfolded, some proteins are synthesized in excess. So all those proteins are degraded in the proteasome. So proteasome helps in protein degradation. So once all these excess proteins are degraded in the proteasome, this proteins are degraded into small peptides. Now these small peptides are transported by TAP. TAP is an ABC transporter, so it uses energy from ATP hydrolysis to transport peptides into the MHC molecule. So basically the MHC class 1 molecule will be bound to your self peptide and not any peptide which is coming from the outside. So initially during the stabilization of MHC class 1 molecule, it is very important to note that the MHC class 1 molecule stabilization is brought about by self peptide and not peptides from the exogenous pathogens. So as the peptides, as your own cells peptides are transported by TAP, so it will again further undergo several proteolytic cleavages by the enzyme ERAP. This is again causes, this is an exopeptidase, and this will also cleave this peptides, which are larger peptides, into smaller peptides. So it will help in cleaving the larger peptide into smaller peptides, and this small peptide will then bind to MHC class 1 molecule. Now, once the peptide binds to MHC class 1 molecule, all the chaperone, that is calreticulin, tapacin, ERP 57, etc., everything will be dissociated from the MHC1 complex, and it will go and it will just leave the complex, and here the class 1 MHC molecule becomes stabilized, which can exit the rough endoplasmic reticulum and express on the surface of your plasma membrane. So this is the pathway how class 1 MHC molecule is stabilized and assembled. And MHC class 1 molecule, again, just to repeat, MHC class 1 molecule can bind to TC cells or the cytotoxic T cells, and also bind to the CD8 plus receptor, co-receptor. Now, the next pathway that we can talk about is the exogenous pathogen processing pathway, which is mediated by the MHC class 2 molecules. So, MHC class 1 is mediating endogenous pathway, that is pathway associated with viruses. MHC1 is endogenous pathway associated with viruses, and MHC class 2 is exogenous pathway that is associated with bacteria or protozoa, fungi, etc. So, in the exogenous pathogen processing pathway, antigen is present in the outside of your cell and not inside. So, the antigen is endocytosed in a clathrin coated vesicle. In cell biology, I think you have heard about different proteins like coat proteins, which are present on the surface of your vesicles, and they specify the vesicles that where they should go. So there is clathrin proteins, COP1 protein, COP2 protein, etc. So clathrin proteins are associated with antigens engulf with endocytosis of extracellular proteins. So antigen is an extracellular protein for the cell. So here it will be engulfed by a clathrin coated vesicle. This clathrin coated vesicle will then fuse with an endosome to become an acidic vesicle whose pH will be around 6 to 6.5. And in this endosome, the antigen will be broken down into smaller pieces. So antigen is cleaved and processed into smaller pieces. Here again, once the antigen is cleaved in smaller pieces, the antigenic receptor will be recycled, and again it will be expressing on the surface of your cells, antigen presenting cells. So MHC class 2 is present on the antigen presenting cells. So these are this is the surface of say dendritic cell or B cell or a macrophage. So which are antigen presenting cells. So yeah, so back to where antigen is again processed and cleaved into smaller fragments. So these fragments are again present in form of a vesicle. Now, this vesicle containing antigenic peptides or fragments will fuse with a vesicle containing MHC class 2 molecule. So, MHC class 2 molecule, after being synthesized in the endoplasmic reticulum, it is going to the Golgi apparatus and is packed in a small vesicle. So, MHC class 2 vesicle, in MHC class 2 vesicle, why MHC class 2 is stable? Because any kind of MHC molecules is unstable whenever they are in a peptide unbound state. But here why MHC class 2 is stable? Because it is again bound to another peptide, which is known as the invariant chain. We will talk about the invariant chain. So what happens is the antigenic peptides which are fragmented, this will go and bind to the MHC class 2 containing molecules, and here MHC class 2 molecules will bind to your smaller peptides by removing the already bound peptide in the MHC molecule, and ultimately the MHC class 2 will be expressed on the surface of the cell. So how antigen is cleaved into smaller pieces? Because of the acidic property of endosome, the acidic enzymes will cleave the antigenic protein into smaller peptides. Now, how is the MHC class 2 stable, and how is the MHC class 2 recognizing these antigenic fragments inside the cell? What is happening? So here we can see that MHC class 2 molecule having alpha and beta chain, this will bind to an invariant chain in order to stabilize it. So this invariant chain, once it is present inside a vesicle, it will help the MHC molecule to be stabilized. But when it fuses with the vesicle containing antigenic peptides, this vesicle is basically acidic or having a pH of 4.5 to 5. So this bigger peptide will be cleaved into small peptide, and only a small portion of this peptide known as clip will remain attached to the MHC molecule. Now, if clip is attached to the MHC class 2 molecule, the antigenic peptides will not be able to bind. So what happens is, this is the digested invariant chain due to these acidic enzymes. So in order for the peptides to bind, clip has to be removed. So how is clip removed? There is another MHC molecule known as HLA-DM, which is shown here as plus. So the HLA-DM will bind to MHC class 2 molecule, and then it will allow the opening up of this particular domain, and opening up will result in the release of clip. And as soon as clip goes away, there are several peptide fragments, so one of the peptide fragments will bind over here, and then it will result in the recognition of antigen by MHC class 2, and finally MHC class 2 will be expressed on the surface. So HLA-DM and HLA-DO, these are two proteins which help in the positive and negative regulation of the MHC class 2 binding with clip, and it is essential for their activity is very much essential for peptide binding to MHC class 2. Now there will be a brief comparison between the exogenous and endogenous pathways. So let us summarize what we have studied. So in case of an endogenous pathway, it is initiated by class 1 MHC molecule. So there is an endogenous antigen. Endogenous antigen, it is cleaved by proteasome. And this proteasome cleaving will result in presence of smaller peptides. These peptides will be transported to the rough endoplasmic reticulum using TAP. Once it is transported to the rough endoplasmic reticulum, we have our MHC class 1 complex, which is bound to several chaperone proteins. But this endogenous antigen, as soon as it binds to your peptide, all these chaperone proteins gets removed. So class 1 MHC chain will bind to calnexin, ERP-P57 and beta-2 microglobulin. Calnexin dissociates, calreticulin and thalassin binds as MHC captures the peptides. So as MHC captures the peptides, all these particular chaperone proteins will release and dissociate. So the peptide is now bound to your MHC molecule. Now once the MHC molecule is bound to your peptide, it will go to your Golgi complex, and from that Golgi complex, it is transported to the plasma membrane, where it is going to be expressed as a class 1 MHC molecule. In the exogenous pathway, what will happen is, there is an external antigen that will be present in your present in the cell. Your MHC class 2 molecule is already in your rough endoplasmic reticulum, which is bound to an invariant chain that will prevent the that will basically block the binding of endogenous antigen. And this endogenous and this class 2 MHC molecule will then be removed. The invariant chain from the class 2 MHC molecule will be removed once there is any kind of antigen, extracellular antigen in the blood. So class 2 MHC molecule is routed through the Golgi and endocytic compartments, where it will come across with the antigen from exogenous antigen from outside. And once the exogenous antigen from outside is processed and present in forms of small peptide, it will go and bind to your MHC class 1 molecule, here class 2 molecule, and here your invariant chain will get dissociated because of the acidic properties, and just the clip will remain attached to MHC class 2. Once the MHC class 2 is attached to clip, there is another molecule called HLA-DM that will bind to MHC class 1 and release clip, and therefore this small antigen peptide will bind to your MHC class 2, and finally the MHC class 2 will be expressed on the surface of your on your cell. So these are the two summary of exogenous and endogenous pathway of class 1 and class 2 MHC molecules. Now we will be solving certain questions. If you have any doubt, you can ask me now. Otherwise, we'll move on to questions. Okay, so I'll be setting up a poll, and also the question will be displayed on the screen. So uh, tell me the answers of this question. You can answer in the poll.
Ma'am, D option, ma'am.
Hello? Yeah,
D option, ma'am. Second one, ma'am. Answer.
Second one.
right, right,
Yeah.
right. So the T cell receptor can bind to the antigenic peptides only when it is loaded to the MHC molecules. That's the correct answer. The next question is this.
A is the answer, ma'am.
No, A is not the answer. MHC2 molecule is present in rough endoplasmic reticulum, but it does not bind to an antigenic peptide. Why?
Ma'am, C option, ma'am.
No. So what does MHC class 2 molecule is initially bound to when it is being synthesized? What is present over here? There is something that I was talking about, like a big protein, which helps in giving the stability to MHC class 2 molecule in its vesicle. So basically MHC class 2 has an invariant chain that is bound in this in the peptide binding cleft initially, and that will allow the that will give the stability of MHC class 2 molecule in its in the vesicle form. And because this invariant chain is present, so the antigenic peptide that is coming from your pathogen, it will not be able to bind initially. Later on, when this protein is removed or dissociated, then only it will be able to bind. So the correct answer will be option number D. So here when I was talking about the invariant chain, so here you see MHC class 1 molecule is bound to an invariant chain. So if it is bound to an invariant chain, it will not be able to bind to this red dots or red peptides. It's going to bind to the red dots or the peptides only when this small protein is removed. So this is known as, small protein is known as Clef, and the bigger one is known as the invariant chain. So this protein needs to be removed in order to bind to your antigenic peptides. So initially, if your protein is having MHC molecule is bound to, is basically the binding groove is blocked by this invariant chain, so it is not, it will not be able to bind to the antigenic peptide. Did you get it?
Yes, ma'am.
Okay. So let's go to the next question. So what is Mhc restriction? Anybody knows?
Ma'am, D option, ma'am. Both MHC1 and MHC2.
CD4 plus T cells will recognize just one MHC. Which is that? This is T helper cells. T helper cells recognize which cell, and T cytotoxic cells recognize which MHC. Before that, do you know what is Mhc restriction? Anyone? Can feel free to tell. It's not a problem if you don't know. You can try once.
मैम जो एमएससी को ही रिकॉग्नाइज करेगा, मतलब जो
Hmm.
बस एक ही एमएससी को ही रिकॉग्नाइज करेगा, जैसे कि टी सी है
Hmm.
वो एमएससी 1 को ही रिकॉग्नाइज करेंगे, वो और
Hmm.
दूसरे को नहीं करेंगे।
करेक्ट। तो हाँ, एमएचसी क्लास 1 को ही रिकॉग्नाइज करेगा, उसको भी रेस्ट्रिक्शन बोलते हैं। और रेस्ट्रिक्शन मीन्स, द टर्म रेस्ट्रिक्शन मीन्स दैट इट हैज टू रिकॉग्नाइज बोथ द टी सेल रिसेप्टर and your CD, like whatever the CD4 or CD8 along with the peptide binding cleft. So MHC class, any MHC molecule CD4 plus or CD8 plus, so any MHC molecule ऐसा दिखता है, राइट? तो उसमें ऐसे पेप्टाइड बाइंडिंग क्लेफ्ट में पेप्टाइड रहेगा. Here one T cell will bind. So MHC class restriction means it has to recognize, and here will be some CD4 plus or CD8 plus. The Mhc class restriction means this T cell receptor should recognize both the peptide binding cleft, the Mhc component where it is directly interacting with the T cell receptor, as well as the CD4 or the CD8 co-receptor should bind. So all three needs to be satisfied in order to show like proper binding. That is why it is known as restriction, because if any one of it is not binding, then the binding between the T cell receptor and the Mhc molecule will not be completed. So that is what is Mhc restriction is. So what kind of co-receptor does Mhc class 1 and Mhc class 2 show? Or what kind of cells does Mhc1 and Mhc2 interact with? So MHC class 1, most of you, like MHC class 1 is interacting with T cytotoxic cells. So this is MHC class 1, and T helper cells interact with MHC class 2. So the correct answer is option number C. Okay. Now let's go to the next question. This one is an easy question, and we have been discussing it for a long time. Right. So the correct answer is option A. So MHC class 1 is expressed by all nucleated cell, whereas MHC class 2 will be expressed by B cells, dendritic cells, macrophages only. So the correct option is option A. The last question for today. So CD4, what is CD4? It's a co-receptor. Okay, co-receptor which is present, which interacts with T helper cells. Like, and what T helper cells interact with is MHC class 2 molecules. So the correct answer is it binds to constant regions of MHC class 2. CD4 is not a heterodimeric molecule. It's just having one domain, so it's homodimeric. It's not dimer also. I think it's just a monomer, CD4, as much as I know. And also the option C is not correct. It does not. So processed peptide will be bound only by an MHC molecule, not by any CD receptors. So CD receptors, these are not binding to any processed peptides. Processed peptides binding is only mediated by MHC molecule, and this MHC molecule will then go and bind to a T cell receptor. CD4 and CD8 binding, always remember CD4 and CD8 binding is no way related to this direct interaction between antigen and antibody, or antigen and T cell receptor. It is just a co-receptor. It is what are co-receptors? Co-receptors are nothing but just that are playing an accessory role. Here is the peptide, here is the, can you repeat the constant part of MHC2, ma'am? Constant regions of MHC2 are these regions. So constant regions are these regions. So these are the regions which are constant in case of MHC molecules, like more or less constant, and these are the variable regions. So all the membrane proximal domains that are not directly interacting with your peptide, those are constant regions, whereas the regions which are interacting with your peptide, these are the variable regions. This is the constant. Okay. And CD4 will interact with, so CD4 will interact here in this way. CD4 interacts with this constant region. Here there will be a peptide that will interact with a T cell receptor over here, just in this way. So peptide binding group will contain a peptide which is directly interacting with an MHC molecule and a T cell receptor. So CD4 is not going to bind to any processed peptides, or it has no role in binding to this direct binding to T cell receptor, or direct binding to a processed antigenic peptide. So it is just a co-receptor. It is just helping the binding to be much more stronger and specific. Apart from that, there is no such direct interaction. That is it. I will go through the messages once to see if anybody has any other questions. Kumar, you have a question? Rajkumar?
Hi, ma'am.
Yeah, hi, yeah. You have a question?
One second, ma'am. Fourth assignment, one question. Second question, doubt, ma'am.
Fourth assignment, second question.
Second question, ma'am.
Okay, what is the
Junctional diversity refer to.
one second? What? Can you tell? Can you tell me the question? Okay.
Junctional diversity refers to the.
Hmm. Junctional diversity in T cell receptors, or what is the question?
Yeah.
Okay.
This question explanation, ma'am.
Right. So let me go back to the slides that I had shared. We were like discussing about how variability is brought about in different kind of receptors, right? So one could be VDJ recombination, and then different how different kind of segments are coming together. So in that case, what happens is, like in case of junctional diversity, this nucleotide sequence of RSS is recognized by RAC proteins and all. And after recognizing these sequences, what happens is there will be some kind of like random addition of nucleotides in between the two segments, like this. Between this base pair sequence, the spacer sequence that we have between monomers and heptamers, that is like 23 base pair or 12 base pair, right? But what is the sequence? That sequence is not conserved. So like heptamer and monomer, these sequences are conserved, but this 23 base pair spacer sequence and 12 base pair spacer sequence that is present, that is random. So this random addition of nucleotides between these two segments of any kind of receptor in B cell receptor or T cell receptor, that is providing junctional diversity. That is what is providing sequence diversity in case of both T cell and B cell receptors. Hello, Rajkumar.
Tell
Yeah,
me, ma'am.
yeah. So did you
Ma'am.
so yeah. So the junctional diversities question that you are talking about. So that was like what is junctional diversity is the question, right?
Yeah.
So junctional diversity takes place because when this heptamer nonamer sequences, they have like some base pair spacer sequences, like 23 base pair or 12 base pair spacer sequences.
Okay,
So
ma'am.
these spacer sequences that are present, these are added randomly by this TDT polymerase, or the polymerase that is randomly adding nucleotides after you recognizing this particular sequence. So these sequences, 23 base pair and 12 base pair sequences, they do not have any sequence conservation. So they will be very different in different kind of segments. So this is providing junctional diversity.
Okay,
Yeah,
ma'am. Ma'am, answer one more question. Answer.
yeah. Which question?
Second question, ma'am. Junction
Mhm.
diversity or answer. Random addition and deletion nucleotides. Option
Yeah.
A, correct answer, ma'am.
Yes, so that is like random addition of nucleotides or deletion of nucleotides in this, like randomly it is being added. It is not added in a template dependent manner. So when these two sequences they are coming together, when they are recombining, say, so that time what will happen is it will not recognize any kind of template. It will add the nucleotides in a template independent manner. So TDT
Okay.
polymerase, terminal deoxynucleotide polymerase, it is adding nucleotides in a template independent manner. So as a result of that, there will be random addition of nucleotides without any kind of sequence specificity. You see?
Okay, ma'am. Uh, Same question, exam question, one more question. Says
Okay.
random addition only option. A option.
Huh. Okay. So what is the like? What is the question? So the answer is option A only, or what?
Okay, ma'am.
Week four.
Ma'am, once again, ninth question, ma'am.
Week four questions, right? Antibody diversity results from this. No, no, no. Junctional diversity. Random addition and deletion of nucleotides between the junction of immunoglobulin. Yes. So when it is adding, so it can like when it is adding the nucleotides, there can be some kind of like it is not very stringent process of adding the nucleotides. It can delete us. So it is like it is not having any template to be specific. So any nucleotide which is added, it can also be coming out.
Okay, ma'am.
Yeah, that is why it is written deletion. It is not like very specific nucleotide addition.
Uh,
Hmm. So what question next?
ninth question, ma'am. The fact that
Okay. The fact that RBC does not express Mhc molecule is well exploited by Plasmodium, entamoeba, leukemia, typhoid. So, yeah, Plasmodium is the parasite for malaria, right? Plasmodium life cycle is in the RBCs initially, and then it goes to the liver cells. So RBC. So if you are like so, uh, let me go to. So if your RBC molecule is not expressing any kind of MHC on its surface, so any pathogen which will target this RBC, so it will not be cleaved because for T cell activation, we need MHC antigen presented with an MHC molecule. Now RBC do not have an MHC molecule, as a result of which any pathogen that will attack an RBC cannot cannot present its antigen to any kind of T cell. So RBC pathogens that are attacking RBCs, these are mainly removed by macrophages or dendritic cells because they can recognize PRRs present on this membrane, PRRs or pathogen recognition receptors. So pathogen associated molecular patterns can be recognized by PRRs which are present on surface of macrophages or dendritic cells. So basically one part of your immune system, that is the uh, dendritic cell. So if it is not able to present its antigen to the T cell, then your adaptive immunity is not going to work in its specific term. It's not going to work. So if adaptive immunity is not working, it will just the pathogen will be tried to remove by this innate immunity by macrophages and dendritic cells. It will take a lot of time. So here pathogen acts as a smart thing to attack RBCs. So RBCs will not be able to present its antigen, and so there will not be any kind of antigen because specific immunity against this plasmodium. Did you understand? This was the question, that fact that does not express MHC molecule is well expressed by plasmodium. And rest other options. Entamoeba is causing amoebiasis. It is not attacking RBC. It is attacking your gut cells. Leukemia is cancer cells, so it is again not RBC. So like leukemia is attacking RBCs only, but it is a cancer, like it is a cancerous form, not any kind of pathogen. Typhoid also, it is not attacking RBC. So Mycobacterium typhi is not going to attack RBC molecules. So the correct answer will be Plasmodium. Did you understand, Rajkumar? Okay, Datta Bhushan and Shashwata, you have questions. So can you go ahead with your questions?
Good evening, ma'am.
Good evening.
Ma'am, question number 5. When IgG bind to protein A,
Question number 5, right. So IgG binding property of protein A is exploited to what kind of thing? So, right. So what will happen? So what happens is the Fc region of your antibody. Okay, so Fc region of your, this is suppose an IgG molecule, and it is having an Fc region. This Fc region will bind to protein A of Staphylococcus, say. Now, so this is non-specific binding. This Fc region can bind to the protein A of Staphylococcus by non-specific binding. It's not any kind of specificity. So, in any kind of chromatographic columns or beads, you have this beads here. If you are just uh, you know uh, taking this part of your protein A component of your staphylococcus and adding on top of your beads, like your beads is coated with protein A on top, protein A on top, all over the bead there is protein A. So what will happen is when you are purifying any kind of antibody, say IgG or IgM or IgD, so it is going to bind to protein A. So the Fc region will bind to the protein A. So what will happen is during purification, all these IgG molecules will be bound over here, because IgG's Fc region can interact with protein A. Now, as IgG is bound in these beads, then you can elute IgG by several kind of buffers that can, be specific to IgG, like specific has a specific pH or specific ionic properties to elute IgG. So this property of protein A binding is here. It is kind of exploited in order to purify this particular protein. So any kind of this gel purification or bead purification, affinity based chromatography that we have. So certain, in affinity based chromatography, you have certain kind of molecules that are coated on the surface of beads, so that it can interact with the other proteins and can bind to the other proteins and elute, and then we can elute the protein of our interest with specific buffers. What is M2C, ma'am? Biobenzene. So you have asked what is M2C. So M2C is the MHC class 2 containing vesicle. So that is just abbreviated as M2C, MHC class 2 containing vesicle. Okay. SFJ, not all the MHCs recognized. SFJ, not all the MHCs recognized by both CDS. Can you repeat the question? I did not understand. What is CDS? Complementary determining, what is that? Okay, it's not the question. Okay. Okay then. So nobody has any other questions? Answer one. Okay. Okay, SFJ. Okay. Cool. So if that's all, if nobody has any other questions, we can say goodbye for now. Have a nice weekend. We'll meet again next Friday, 7 p.m. So thank you, everyone.
Thank you, ma'am.
Thank you.